Hello, everyone, and welcome to today's live webinar, which is being sponsored by Benchling. I'm Jeff Bogaliskis, technical editor for Genetic Engineering and Biotechnology News, which has been the leading source of information on new tools and technologies within the life sciences industry for over 40 years. So many of you, I'm sure, are well aware of the essential nature of in vivo studies for the drug development process. In vivo work is critical to helping scientists and clinicians understand new compounds' pharmacology, toxicology, and overall safety profile. Due to the critical aspects of knowing this information, it's imperative that rigorous data collection be performed. Yet many teams still struggle to manage the end-to-end process of running in vivo studies using spreadsheets, emails, and outdated point solutions, leading to laborious data collection, transcription errors, and poor collaboration across teams. Now, modernizing these in vivo workflows with novel cloud-based informatics solutions is how many organizations are pushing ahead at a more confident and faster pace. And our presenters for today's webinar, who you'll meet and learn more about in just a few moments, are going to tell us more about the novel informatics solution called Benchling Studies and how it can help accelerate your in vivo work. But first, I have a quick reminder for our audience members. We would obviously love to hear from you during this event, and we'll be taking questions toward the end of today's presentation. So if you have any questions for our presenters, don't hesitate to send them in at any time during the event. Just type your question into the Q&A section and hit submit. And we're going to try to get through as many of your questions as possible. Okay, with that said, I'm going to turn the presentation over to our first speaker, Dr. Chris Armstrong, who is strategy lead at Benchling. Chris, over to you.
Yeah, thanks, Jeff. So to get started, I just want to start by doing some introductions into our speakers today, of which you'll see that we have three. So first of all, to introduce myself, my name is Chris Armstrong, and I'm a strategy lead currently with Benchling. But before this, I was the CEO and co-founder of a company called Overwatch Research. This was a company that was acquired by Benchling earlier this year. Prior to this, I had extensive background working within in vivo research settings, and I continue to lead this function within Benchling. We're also joined by Wendy Fernandez-Ochoa, who is a scientific solutions consultant at Benchling. And previously, she was a scientist at renowned institutions such as the Burnham Institute and Scripps. And we're absolutely delighted to be joined by Catherine Souza, who is a senior in vivo research associate at Servier, a leading global biopharmaceutical company who has worked extensively across the in vivo function. And so with that, let's get started. Now, the theme of today's webinar is to discuss how we can modernize in vivo R&D programs. And over the next 10 minutes or so, I'm going to provide you with some industry perspective, specifically focusing on why it's imperative to align in vivo programs with modern R&D informatics. I will then pass to Wendy for a demo of the Benchling Studies platform, which is our connected cloud solution for in vivo R&D. And then finally, as I say, we're delighted to welcome Catherine Souza from Servier, and we'll have a short fireside chat to discuss experiences with in vivo study management. And just another reminder, please feel free to send through questions at any time, and we'll try to answer as many as possible at the end. So this statement will not surprise many in this audience, but I think it's important to frame the problem that we are collectively trying to solve. The number may vary slightly depending on therapeutic area. However, it's widely accepted that approximately only 12% of drugs that enter clinical trials are ultimately approved for use in the clinic by the FDA. Now, this represents a colossal waste of resource, but more importantly, a failure to help the patients that require innovative new therapeutic intervention the most. And so why do nearly 90% of drugs fail to gain approval? Well, there are many reasons that we don't have time to go into today. However, I do want to focus on some that I think can be related back to the preclinical phase of evaluation. It would obviously be ideal if we could identify things like lack of efficacy or issues with safety prior to these drugs entering into expensive clinical trials. After all, isn't that why we perform in vivo research in the first place? And so if both of these could be tied directly back to unreliable preclinical data, where this is much of a bigger problem than people are generally aware of. And it's essential that we have 100% confidence that the preclinical data that we're using to justify clinical trials in the first place is as robust and gives us the confidence that we need to take these forward. So just how big of a problem is this? Well, this well-known publication highlighted the prevalence of irreproducible preclinical research exceeds 50% and costs over $28 billion each year in the USA alone. Now, this was reported in mainstream media around the world and was framed in a way that I personally experienced throughout my own research career. How many times has everyone attempted to reproduce findings of another research team or your colleagues, or even data that you previously generated yourself, only to be surprised to obtain totally different results than expected? Fortunately, you're not alone. However, we do need to start taking action to ensure that this problem does not persist. So which areas of our research process require improvement? Of the over $28 billion wasted every year on what is essentially bad science, over 50% could be attributed to things like improper study design or inadequate data analysis and reporting. Now, we're incredibly fortunate to live in an era with never seen before technological capabilities. These are problems that we are better positioned than ever to solve. However, we're not going to do this by relying on spreadsheets and paper-based laboratory notebooks. It's crucial that we're using more advanced software tools to improve our research process and the data that is subsequently produced. Now, the focus of this webinar is, as we say, on in vivo research. So how do the stats line up? Well, I'm afraid to say that they're actually even worse. If we focus specifically on data generated from animal studies, our ability to reproduce falls to as low as 11%. Is it surprising that so many drugs fail clinical trials when we can only have confidence in 1 in 10 of the studies used to justify progressing in the first place? So this is something that we've been deeply interested in for a number of years now. And the main gap that we observed is that there is no cohesive option for companies that wanted to manage core in vivo functions as well as the connections to both upstream and downstream R&D teams. Now, on this slide, you'll see 4 of the primary functions that we routinely see our customers asking for. It's typically things like study management, colony management, study and assay documentation, and also downstream bioanalysis. Now, there's often a high degree of interaction with free transactions needed between these. For example, to transfer animals from a colony system into a new study, or transfer samples collected during your study to a downstream team that will perform bioanalysis. And many of our customers were typically using things like spreadsheets and email for this study management, which required a lot of manual data entry and made it difficult to access data across the team. Some were even using point solutions for things like colony management, but this too created a major silo and required a lot of manual data transfer. Other informatics tools such as ELN or LIMS can also play a role here, but also have limitations when used as isolated point applications, especially in a world of modern drug development, where these tools are often not ready to handle therapies or samples that require experimental, scientific, and/or sequence context. And so what we've observed across our 900 plus customers in the biotechnology industry is that drug development has generally become faster, more organized, and more collaborative across most functions. In vivo research is an essential component of drug R&D and requires the same level of modernization. Otherwise, it risks becoming a bottleneck for our R&D programs. Many of the investments that biopharma companies have made into their R&D technology and software haven't yet made their way to in vivo teams. And we typically see that this is the case because in vivo studies require a great deal of functional specialization to enable you to do things such as managing your colonies, randomizing animals into studies, or even tracking in-life data and so forth. So historically, these teams always go back to relying on spreadsheets or even heavily customized in-house applications that are generally difficult to use and difficult to maintain. The truth is that these in vivo teams require more specialized solutions and one that supports their key workflow needs while reducing errors, saving time, and increasing collaboration. It's therefore critical that in vivo programs not only have a solution that directly supports study management, but that also helps to connect their work to other R&D teams that help advance programs using this data. And that's why I'm delighted to introduce Studies, which is Benchling's application for in vivo study management. As part of the Benchling R&D cloud, it is designed for accurate, intelligent data capture, team collaboration, and insights that can be used to advance drugs into the clinic. Importantly, Studies was developed by experienced lab researchers who knew firsthand how difficult it can be to manage data across their studies. The product was designed to help modern biopharma R&D teams get their studies up and running as quickly and as accurately as possible. And in Vivo teams can use the application to capture all of the required in-life data with a focus on automated data capture to reduce manual data entry and transcription errors. And importantly, as I said, this is part of the R&D cloud, which means that studies can be integrated to the rest of your Benchling applications to create connected, collaborative workflows across your teams. So let's take a closer look at what each of these core areas means and how it might apply to your in Vivo R&D. I'm sure that many of you will be familiar with things like colony management systems. Many of you might have even been using them this week. And they offer the ability to track animals that pass through your facility, allows you to assign information to those animals, and even has some specialized functionality, such as the ability to print off custom pH labels. However, these solutions often exist in silo, with no connection to the other systems that you use as part of your research process. In the Studies product, we have recognized the importance of connectivity and enable colony management capabilities that feed directly into the study creation process itself. We've also made it as simple as possible to randomize animals within your study, ensuring that your process is not affected by what is unconscious bias. Users can easily apply automated exclusion criteria prior to commencing with multi-parameter randomization, ensuring that you can randomize on the basis of several factors simultaneously. And we also put a lot of focus on automating many of the repetitive tasks that are performed on a day-to-day basis via integrating with commonly used laboratory equipment. And this includes things like RFID scanners for identifying the animals, balances for recording body weights, or even calipers that can be used for things like measuring inflammation or tumor sizes. We can also provide a mechanism to easily import data generated from more complex devices, such as imaging systems. Now, to tell you more about these exciting features, I'm going to pass you to Wendy for a product demonstration. Over to you, Wendy.
Thank you, Chris. So I'm going to jump into the platform in a minute. But before I do that, I want to give you an overview of what the main three subjects that we are going to talk about. We are going to talk about centralizing and standardizing data capture. We are going to talk about streaming tasks and reducing inefficiencies. And we are going to also cover ways in which you can improve collaboration, both internally and externally. I have logged into Studies. Studies, as you have heard, is a cloud-based platform. And so through a modern browser, and here you can actually access your account in Studies from any device you have connected to the internet. So this is my account, and permissions can be set up at the level of the account itself and also at the level of a study, as we are going to see in a second. So one of the main challenges we hear from people is the idea of centralizing all this content related to all the studies they're running, sometimes at the same time. And so as I get into my account, what I can see is all the different studies that I have access, that I belong to. So I have my active, drafted, completed, canceled studies. And here I can see also who in my team is participating and in which level or which role. Maybe I'm an author, but I have other people collaborating, maybe as writers or just also reading only options. When I want to learn more about in a study in particular, so I can get into all the information that is relevant to that study, organized around all these different tabs. So you can see that from here I can access the summary, all the different metadata about the study, but also information relevant that I'm aggregating to the study as it's created. So I have all these attachments from PDFs, documents, images, all in one place. I also have information about the cages that are housing the animals, also the animals, observations, the treatment groups. All the information is going to be centralized in one place. I'm here in my panel where I can see all the animals that are involved in my study. And right away, I can see not only the list of the animals, not only the case, the cage, but also metadata like the age, the tracking date, the study group they belong to, and even measurements that I've been capturing for each of the animals. Here I can see the last measurement that I did and the difference between this measurement and the first measurement we took. For this particular study, we are measuring tumor volume and weight of the animal. Also in this list, what I can see right away is these alerts. These alerts are telling me that some of these values had changed above a certain threshold or above like certain percentage. And I have set up the system to notify me for these particular changes. So I can be alerted of things that are happening with individual animals. So I'm not surprised that something happening to an animal later. For each of the animals, I can have this information and more information when it comes to all the different measurements, and I can follow the progress of each animal at the level of each of the measurements we are taking. Again, for this particular study, I just have 2 measurements, but the amount of measurements and the type of measurement would be completely up to you. It will set up at the administration level, and then you will have flexibility of what is that you are collecting for your particular study. Also, what I can see here, observations. So as I collect information about those animals, all the information is actually captured here. Here I have also the samples. I have collected different samples from this animal, and everything is aggregated to the animal itself. Doses, I have information about what is the treatment that I'm associated with that particular animal. So you can see that I can follow the animal one by one. But usually what I want to see is how am I following the study in general. So here in this other tab, what I can see is how the study is progressing at different levels. You can see here the 2 different measurements, and I can see like how the groups are progressing and the groups are changing with all the measurements that we have taken along the progress of the study, including a Kaplan-Miller survival curve here to give you information about how your animals are doing. So as you have seen here, for each of them animals, I can actually go here and collect information, collect measurements as I need to for each of the animals. In reality, this could be one of the most tedious tasks in a study. So in studies, what we have is what we call a workflow that is going to help you streamline the data collection. I think the best way for me to show you how this works is actually show you a video. In this video, what you're going to see is somebody simulating, is a scientist is taking each of the animals, scanning the tags, so the system is recognizing the animal by the tag, using a caliper that is connected through Bluetooth, and also capturing the body weight by balance that is also integrated with the system. As the scientist is collecting this data, the data is added automatically to the system and is updated live in the system. So this is what a workflow will look like. And again, as you connect things and you go through the process, that information will be updated live. So it's not only me that I'm collecting the data, that I can see the progress of these measurements. Is anybody that has access to these studies collaborating with me with this access will have real-time information about all these measurements we are taking. So another way to actually import information into the studies would be in bulk. So imagine that you are using an instrument that produces a particular type of a tabulated file with the data. You can always bring this information associated with the day you collect the data, drag that file into studies, and all you have to do is confirm that the columns, the information in your file correspond with the information you are capturing in studies, and that information will be automatically aggregated to those animals and that information will be, again, updated live and visible to other members in your team. So when we talk about animals in studies, we have kind of three main, I'm going to go back to my studies, three kind of stages for the animals. So when we come to the colony management, so these animals, here we can see all the different animals that we have, and some, as you can see, are associated with the study, and some are not yet associated with the study. So one of the steps for us would be to associate certain animals to the study. Once those are associated with the study, those animals will show up in our animal panel. What you can also see here is some of these animals are associated with a group already. So animals can be in the study without being associated with a group, or associate those animals to a study. So what I want to show you now is how you would actually associate those animals to a study, so to a group inside the study. If I come to my animals, you can actually, yes, absolutely, you can manually associate those animals to a particular study. But what we have is an efficient way to eliminate any bias from your associated to a groups. So you can see here, what I'm going to do is actually use the randomization process. I'm selecting 25 animals that I want to associate to study groups, and I'm going to use the randomization process to associate those with the groups. I have selected, or I have told the system that I need 5 animals per group. Because I selected 5 animals, the first step would be for me, over this system, actually, to exclude 5 of those animals. These can be done based on the different measurements that you are collecting and associated with a particular value for the mean. You can see here that the system is excluding 5 of those animals. And as I move along, so now what the system is going to do is randomize the other 20. And we have options to pick a particular attribute, maybe is sex or the donor ID. And we have up to 3 different metrics that will be used for the randomization. In this case, I'm going to use the 2 that I have. And the process works, and now these 5 animals are associated with each group. So each group is actually have the number of animals we told the system. And here is the randomization has been done, and all these animals that I selected are associated with the different groups. One thing that I could do now is actually recage the animals based on the groups. And you can see that I could in bulk recage all those animals and control the how I host those animals. Great. So the third portion of the presentation I wanted to talk about today is collaboration. So I'm going to assume that we are at a point in which we actually have to share information with maybe collaborators outside or maybe inside, or there's a point in which we need to share information about the study, how the study is, or we want to bring information of the study to another, maybe statistical software. So in export here, you have different options and different formats in which you can actually export different type of information from your study. Another option for collaboration would be a point in which you have samples that you have collected for the animal. And give me a second. So this is my study in which I actually have collected information from the animals, and I have collected also samples. So you can see that I have these different samples here. So, and I want to pass that information and these samples to the teams downstream. So yes, I could actually go and send somebody an email, go tap them in the shoulders, but the idea would be that we are working in an integrated way. So everybody has this information centralized, and this information is flowing without us missing information or making mistakes about how we share information. And so what we have done is studies is actually integrated with Benchling. So if we have our team actually also collecting information in Benchling, what we are going to see is that information from studies is actually automatically transferred to Benchling registry. So I'm now in Benchling. This is a look of the registration system, the sample management system in Benchling. And I have here selected and filtered by all the animal samples that I have in the system that I have transferred from. I have collected in the studies and I have transferred, or the system has automatically transferred into Benchling. And if I'm looking for a particular sample, so you can see like that sample that we actually collected in studies is now here in Benchling. And as I go into that particular sample, I can see also the information from studies has been transferred, not only the ID, but also the study group, the study. I have a link to the study, information that comes from a study as well, and also information that my teams in Benchling have been collected about studies. So all the information about this sample, no matter what it comes from my in vivo group or from the downstream group, is actually centralized in one place. So we have done an overview of these three main topics. We have talked about centralizing and standardizing data capture. We have also talked about how you can streamline task capturing data, and these will reduce inefficiencies. And we have talked about also collaboration with teams outside of my own team, outside of my study on Benchling, actually also collaboration with teams that are downstream from me, collecting data on those same samples through the integration between the studies and the Benchling registry. And I'm going to pass it back to Chris.
That being done, I think it's time to launch into the fireside chat. So as mentioned earlier, we're delighted to have Catherine with us today. And I guess, Catherine, it might be a good starting point if you can maybe just do a brief introduction into yourself, maybe a little bit about your background and what led you into your role at Servier today.
Sure. Thanks for having me, Chris. So a bit of background about me. I graduated from WPI, or Worcester Polytechnic Institute, in 2017 with a bachelor's of science in biology and biotechnology. After that, I went over to Charles River, where I was contracted to Sanofi. For about a year and a half, I was doing a lot of animal husbandry work as an animal tech 2 there. After about a year and a half getting into the role and everything, I wanted to explore more technical possibilities. And so I joined Agios Pharmaceutical in Cambridge. I was there for about 2 years, where I gained a new role in technical experiences. I was a study lead. I worked with the pharmacologist one-on-one a lot of times. In 2021, Servier Pharmaceuticals bought Agios' oncology portfolio, and with it, a number of their scientific staff. So that's how I came over to Servier, and that happened about a year and a half ago almost. So here I have been for about a year and a half, and I am the lead for all in vivo services in the US. We have recently launched our new lab space in the Seaport, and so that's where I am now.
Cool. So sounds like you've been involved in vivo science for a good number of years now. So what would be some of the things that you find most rewarding about it, and what kind of impact do you see yourself and your wider team making?
Sure. So I think I speak for the greater community is helping patients. That's always our number one priority in this field. We're in this to help patients find treatments, and following with that, animal studies are really essential to doing that. We can't treat patients without the animal studies we do. And going off of that, because we're using animals, one of my biggest rewards is advocating for animal welfare. So that's a huge part in my role here at Servier, and one of the biggest things that I love doing for animals. They are essential to our to our studies, and they deserve the utmost respect. And so I really enjoy, I feel very rewarded in advocating for them. Impact-wise, I think moving programs and targets forward or stopping the science, sometimes a no-go decision is just as important as a go decision. And so even if we choose to not go forward with a target, we have learned some new biology about that target that maybe we can focus on more in the future once we know a little bit more. So I think those are the biggest impacts and rewards that I have in this field.
Yeah, no, I couldn't agree more, especially on the wider patient impact and also the animal welfare side. So I know from my own experience that whilst rewarding and people work can also be quite challenging. So sort of switching it a little bit, what would be some of the things that you find most tedious or more taxing on a daily basis?
Yeah, I know I mentioned before animal welfare. That is my main priority, making sure that these animals are healthy enough to keep going on study. That's really important for getting credible data. And so I can keep track of hundreds of animals at a time, really. So going animal by animal and making sure that they each are within the parameters that they need, that can be very taxing. But thankfully, Studies now has the alerts platform. And so on one page, I can really go and see each animal and look at their percentages of going up or down in weight, if their tumors are increasing, decreasing, and I have alerts set for specific parameters dependent on my studies. And so I can go in there and in one look just see that these animals are doing okay or what I really need to be focusing on. So I can make sure an animal never suffers or is mitigated as much as possible. So making sure that they're as humane as possible is important. And then another thing within Vivo work, as we all know, they change constantly. And Vivo doesn't always go the way we want. So we have to kind of roll with the punches. So dosing starting earlier than expected, animals not tolerating a dose or needing to be taken down, a lot of that can be very taxing. And then just the repetitiveness of some of the nature of the studies, taking caliper measurements, doing that kind of stuff. And the last thing is running studies in parallel. Each study has their own parameters, schedules, dosing, animal models that they're using that you need to really be able to keep track of. And that doesn't always line up and can be really hard to track. And so using Studies, I have been able to track all of these animals and really keep the studies going. I know I've done about 12 studies at a time. And so doing that, it can really help.
Yeah, so it sounds like you're a very busy person. So what does a typical day in the life of Catherine look like? What type of studies are you typically working on? And what does progression look like from start to finish?
Sure. So I'll start with the types of studies we run. So we will run any studies from growth validation, just, making sure that the cells that we're injecting actually grow, to kinetics, efficacy studies, PK/PD, model development, surgeries. And in those includes, dosing, measurements, mostly body weight and tumor volume, but also including some body condition scoring, some grimace scoring. I've done paralysis scoring before. And so there's a lot of different scoring involved. And then, like was discussed before, just sample collection, making sure it's all tracked. And so in a day-to-day, I actually work with my vivarium is not in-house, it is through a contract. And so I run our vivarium program, but I do have others. We work with Cradle, and so they come in and do some of the work with us. And so I usually try and do as much hands-on as possible, but when I can't, or I'm away on vacation, for example, they're able to step in. And so a typical study just goes, the biology team will take the cells and everything first. And so they will validate using literature, their assays, and their development tools. Once biology is comfortable with their assays, they will bring it to the pharmacology team, which I am a part of. So the biology team and pharmacology teams will meet and sit down and create a study design to run with the goals and what we're looking to validate in this study. Does it mimic the in vitro data, or does in vivo tell a whole different story? In vitro does not always lead to in vivo, as many of us probably know. So once we get a study, for me, it can last anywhere from 3 to 6+ weeks, depending on what type of study, dosing, just a growth validation, that kind of thing. And Along with that, we're collecting tumor measurements, body weight, body condition scores. We're observing trends. I'm making decisions on when to start dosing, when to stop dosing, that kind of stuff. And then depending on the study, we can have any range of animals from like 20 to validate a cell line to 200 for an efficacy or PK/PD. So most of our studies probably average somewhere between 60 to 100. But like I said, I've run up to 12 studies at a time on my own with 3 additional with a partner. And so it can be a lot. And then after we get that data back after a study, the pharmacology team and biology teams will look at the data generated and decide on our next steps. This process keeps kind of going in a cycle until we have enough data to decide whether we want to move forward in an IND or keep going with research on that target. At the same time, our biochemistry team and other teams are also looking at the target, making sure it's viable. So then we take all of that data together and make a package to decide if we want to go forward and make a decision on the target.
Cool. Definitely reiterates that you're a very busy person. So let's switch to the software side of things now. So you're running numerous studies. Each of them could have 100 or more animals, multiple time points, and lots of measurements and data to collect. So can you talk a little bit about the software tools that you have used previously, that you're using today, and what your sort of overall experience was?
Yeah, of course. So up to this point in my 5 years, I've used 4 different softwares. So currently, of course, using Studies, but I have previously used Excel, Provantis, and StudyLog. So I have found that a lot of their interfaces just aren't as clear to follow as Studies is. I can really follow and find what I need very quickly. Many of the options do have Bluetooth or RFID, which is nice, but not all of them do. I personally don't use RFID. I use a tattoo for my sake, but the RFID is very nice to have. And so for Studies, having those applications really makes data collection a lot faster. And then the capabilities that are very important to me are data capture and finding and seeing my data. So seeing those graphs, seeing those alerts and how the animals are trending. That's the most important thing to me. And in Excel and StudyLog, you really can't see that unless you export the data and do some finagling with it. So I find that having that chart in Studies, being able to just, go and look at the groups and seeing those trends is able to help me decide how the study should continue running. And so I have also found in other studies that once I put in some information, it's difficult to change that. Whereas in Studies, I have found that, I'm like, okay, well, we're going to have to start dosing on this day. I can put in that dosing information super easily and we are ready to go. So just making changes and then just working with the vendors. I found, Excel, you kind of do on your own. StudyLog, their customer service interface, I've wanted some different coding for formulas and stuff to use for some of our studies, and it took a long time. But with Studies, I think that you, Chris, have met with me a few times, and I'm like, what can we do for this? And you've been fantastic in just helping me. And I am up to speed and being able to do everything I want to do within a few months on this platform.
Yeah, no, happy to help, and hopefully we can continue that relationship. So I guess moving into the important bit then, so you've talked about some of the software challenges you've previously experienced, but how did that actually impact your work?
Yeah, so one of the most important things in data collection is accuracy. You want to make sure you have reliable, accurate data sets. When you don't have that, you have to repeat studies due to inaccuracy, and that takes 1 time, 2 resources, and 3 more animals, which we don't want to waste any of those things. So I think, as you said, animal accuracy is really low in our industry. And so I think making sure that the interface is as easy to read as possible, making sure you can see these things with these animals and, finding outliers as you're going really makes a huge difference. So I'd say the data collection and being able to see that data right off the bat has really informed our accuracy and just collecting data and finding trends. And then for randomizing, I'm able to make sure that we have the best animals possible. For example, if I see one animal that, isn't losing weight, and the tumor volume is fine for randomization, but their body condition score doesn't look like they'll be able to be in the study, I can exclude that animal and still randomize. I would have trouble with other softwares with that. And so I think that makes it even more accurate. You don't know what that animal is kind of have underlying, what underlying condition they have. And so being able to generate the most accurate data possible. Having really brittle software can cost a lot of accuracy. And so having a strong platform really reduces that inaccuracy and leads to, better trending and getting further in those parameters.
Yeah. So I think that, I'm just trying to think back, we met about a year ago, and that's when your group would have had the first experience with what was Overwatch at the time, now obviously Benchling Studies. So I think you've probably covered, what I was going to ask in terms of initial experience with the software. Maybe if you have anything else to add there, then please feel free. I guess I want to add a bit of a selfish question here as well. Now that we are sort of a year down the line, what should we build next? What feature would sort of be the one that could make the biggest difference to you?
The biggest difference would probably be IVIS imaging and integrating that into the software somehow. We are starting to use IVIS imaging quite a bit more. And unfortunately, the two softwares, we haven't found a good way to mix that yet. And so I know that we've spoken briefly about it before, but I think including those data sets in there would be really beneficial. Right now, it's kind of a long and tedious process, and you can't get all the metadata when you try and, take the images. It has to go into their platform, and then you have to download it, send it, and then upload it to another study or platform for analysis. It loses a lot of the metadata, and it's kind of a tedious process that can cause a lot of inaccuracies or mistakes in there. And so I think if we're able to integrate those two systems, it would help a lot. And again, the credibility of the data would be much higher.
Yeah, no, I totally agree, and that's something that I'm more than happy to talk to you as well. So yeah, I guess on the same theme then. So we're nearly a year down the line now. So what type of impact have you seen studies having within your lab so far? What aspects of the software do you think have added different value propositions to your team?
Yeah, so one of the main things is time. It helps me keep track of many different studies. Like I said, I can run a lot of studies at a time. And so it really saves me time and, again, accuracy, knowing which study I'm going into and what has to be done, X, Y, and Z. And so that has saved a ton of time and resources. Another thing is I said that we worked with a CRO, Charles River, who sometimes does studies. They have their own ID with us. And so they're able to go in, and I'm able to, go and track them. So they'll do the collection, and then I can log into my username and go and check. I don't have to, send an email being like, how did it go, kind of thing. I can go and check themselves. Honestly, I could be there on real time with them if I just happen to not be able to be there in person. So I think that's one of the biggest things that has helped me. And then just data storage on the cloud is fantastic. I can look at it from one location. My boss can look at it from another location. We can email, it emails you directly, which helps a lot. You don't have to be emailing data sets back and forth. Not the most secure thing, but coming straight to your email from the platform is fantastic. And then I personally don't use it, but the rest of my team does. Sending it to Prism for other analyses is a fantastic aspect. So it's just saved a lot of time. And again, keeping everything all compact in one thing so that we don't have to lose any of that metadata is amazing.
Yeah, that's great. So just, one final question then. There might be other people in the audience who maybe in the same position as you were a year ago. And so what advice would you have to other scientists who might be evaluating their software options? How did you go about getting started? What did you consider at the time?
Yeah, so honestly, I reached out to a number of my friends in the industry, see what they were using. And that is how I heard of Overwatch Now Studies. A friend of mine that I worked with was using it and told me to check it out. So I talked to Chris, and Chris was fantastic in showing us kind of what they do, what the differences were, and then let me have a trial period for, I think we agreed on 2 weeks. And I actually ran it in tandem with a study that I was doing on my other software. So I could see the differences between one platform and another. But going about it, I just researched different platforms. What I would suggest is really knowing what you need it for. I knew that I needed to be able to track all these animals and see if alerts were coming up or when dosing was supposed to start, easily seeing trends. And so being able to test that with studies was the biggest selling point for me. It was super easy to use interface, and it really helped me a lot. And so I'd suggest just, doing your research and asking the questions, knowing what you're looking for, and not being afraid to, I think Chris told me, try to break it. So I wasn't able to, and that's a fantastic thing. So yeah.
Cool. Thanks a lot for spending the time to talk to us all today. It's been really, really informative. And I guess now's probably the right time. I can see that there's been a lot of questions coming through. So maybe we should try to cover some of those. So I'll pass it back to you, Jeff.
Yeah, great. Thanks, guys. So Chris, Catherine, Wendy, really great presentations, guys. Thanks so much. We have a lot of questions from the audience, so that's great. A lot of interest. I just want to let everyone know that we have a poll coming up in just a few moments. And so before we dive into the Q&A, if you'd like to learn more about Benchling, the Benchling Studies, or would like a one-on-one demo, please vote in the poll, and Benchling will have someone reach out to you. Okay, great. So with that, let's move on to our questions and try to tackle as many of them as possible. So I'm going to throw these out to obviously the group here, and you guys can chime in as you see fit. So the first question we have, one of our audience members says, we use a range of instruments in our studies beyond balances and calipers, including blood analyzers and the iVis imaging. How does the data coming off these instruments best fit into your software?
Yeah, so happy to take that one. We've seen this quite a lot as we sort of expanded maybe about 2 years ago now into more and more therapeutic areas. Naturally, we were seeing different equipment being used. I think that there's a number of different ways that we can manage this. One of the things that we like to do is within our technical team is to try and understand not just how we can improve your experience with the software, but how we can actually link that in with the environment where you work. So a laboratory is a pretty complex place, lots of different equipment, and we like to usually ask that as part of the process whenever we're onboarding a new company. We will typically see things like blood analyzers. I've seen iVis systems a lot. Like I have previous personal experience of using like micro CT scanners or like small animal radiation research platforms. And I think this links back to what Catherine discussed in one of her answers, whereby it would be absolutely amazing if we could integrate directly into the likes of iVis systems. Now, the reason that Studies is currently not integrated into those systems directly is not because we don't want to. It's generally just because those systems are quite difficult to integrate into, and it's something that we will continue to pursue. And we have had conversations with a number of the companies that provide not just iVis systems, but other imaging systems. The way that can be done at the minute, so as Wendy demoed, we do have an import capability. And so a lot of these devices will produce usually some form of CSV file or even a spreadsheet, whereby we can help you to map that data into the correct format so that you can import it directly against the correct animals within the Studies platform. Other devices, if they do have the likes of Bluetooth or USB connectivity, then we will really work with you to try and see how we can get that to work directly. So really the short answer is that it's taken on a device by device basis, but we're more than happy to have the conversation.
All right. Thank you for that. We've had a couple questions come in on the same topic, but Cheng Ting was the first person to ask this. So I'm going to combine two of the questions together about animal ID and so forth. So they'd like to know how to make animal ID unique and link it to a specific study, as well as how to track animal data before and after assigning to groups. Maybe you guys could talk about that one.
Yeah. Again, happy to dive into this. So within the Studies platform, we can store up to 5 simultaneous ID methods against every animal. Probably a little bit overkill, to be truthfully honest, but it is helpful. And why we did that is that if we went back like 40 years ago in the product, we had a singular animal ID field where you could assign whatever method you wanted, whether it be an RFID, a tail marking, an ear punch. Where we ran into difficulty at that time was we had some companies, just due to their own process, where they might have assigned a secondary ID after they randomized. So they needed the ability to be able to add additional IDs over the course of the study lifecycle. So now what you're able to assign is not just the original ID field. We also have dedicated fields for a tag ID or an RFID chip. an ear ID, a tail ID, and also we have a lot of companies that do PDX type studies. So we also added in the ability to store a donor ID. Obviously, if you're able to avail of RFID, then you do get some benefits in terms of automation, because we can allow you to scan the animals, automatically pull up records. It helps with data collection. But we also appreciate that a lot of teams don't use RFID. And so we've tried to really mimic as much of that automation for the more manual methods as best as we can. So that's sort of covering the ID side. Was there a second part to that question? Sorry, Jeff. Thank you.
The second part was, how to track the animal data before and after assigning it to groups.
Yeah, so data can be assigned to animals regardless of the stage of the study you're at. So that can be before you've randomized, after you've randomized. We have this concept called a tracking date within the software, which can be assigned to animals at any time. And that is effectively the indicator for essentially saying this is the point that we've now assigned these animals into groups. And so what it will do is in terms of your graphing and setting your day zero, if that makes sense, it will only consider the measurements from that point forward, but it will still enable you to store and have all of those measurements captured from before that date.
All righty. Next question we have here, given the sort of, state of the affairs these days with everybody using tablets, cell phones, everything like that, Seth would like to know if there is any plan to support cell phone or tablets via an app or mobile optimized website.
Yeah. So I guess a little bit of a secret that people might not know. Whenever we first started building what was originally Overwatch, it was built mobile and tablet first. Why we did that was actually because, based on my own personal opinion, I thought it would be really cool, probably like the person that answered the question, if I could just pull my phone out and begin to track animal data. Why we stopped that was actually, as we started speaking to more biopharma companies, there were obviously IT departments that had security concerns about scientists accessing in vivo study data on their personal devices. Particularly mobile phones was the use case that they had. However, we are beginning to see more uptake specifically of tablets within vivariums. So we're seeing like dedicated like tablets alongside all of the hoods, and some companies are making that investment. And so we do sort of strive to make sure that we can at the very least offer tablet compatibility going forward.
And if I could cut in here, I actually do use a tablet for studies right now. My company did, set up the tablet, encrypt it and everything. So it is fully safe, encoded, and I have had no issues on getting onto studies / Overwatch and using it on a tablet. It works fantastic.
All right.
Thank you, Catherine.
Yeah. Great plug. Thanks, Catherine. So next question we have, an audience member says, are studies frequently require multiple different injection sites for the animals? Is there a way to track that in the application?
Yes, there is. So we have a treatments and dosing feature. So it sort of enables you to do everything that you would expect. So loading in all of your treatment information, helping to assign different concentrations, and then we can try to do some cool things like automatically calculating dose volumes based on the body weight of the animal. In terms of having different injection sites, there's a number of ways that can be done. So we do enable you to create custom fields that can be associated with the majority of what I will describe as entities in the software. And entities could be things like the studies themselves, your animals, your cages, your treatments. And so you could imagine that alongside your treatments, you could create a custom field, which is simply like a select field or a drop-down field where you call it injection site. And you would be able to have all of the different options that you would want your users to select from there.
All right, great, thanks, Chris. This next one is directed directly to you. Terry would like to know. Says, "Hi, Chris. Can we run two randomizations in the same measured study? Like if we have 100 mice and we use 50 of the mice for efficacy, 10 mice per group, and use the rest of the mice in PK/PD studies, like 7 mice per group. Both studies need to be randomized."
Yeah, and I only know one Terry, so nice to know that you're here, Terry. Yeah, that can be done. So you can randomize as many times as you want with a batch of animals. And that was one of the sort of things that we wanted to achieve with our randomization feature. We didn't want it to be so rigid that it was almost like a one-step process that you had to get right once. And so if you had a situation where you had 150 animals and you wanted to randomize 100 into a set of groups that might be going for a tumor study, and then 50 that are going into groups for another study, then we could effectively run two separate randomization processes with that batch of animals. On the PK side, and I probably discussed this previously with you, Tari, but just for everyone else's transparency, that is something that I have personally and as a wider studies team, we have been looking at for probably well over a year now in terms of doing quite extensive user research and trying to understand the main pain points there. Number of ways that we can address that. And I sort of see two main areas where I would like the product to be able to assist. Obviously on the data management side, which is sort of just, once the study's been done and you have all of that data, how can we present it in a way that makes sense? But I think actually the bigger challenge is the execution of PK studies and how we can help there. So the timing of your dosing, of your bleeds, helping with all of the scheduling, a lot of the unpredictable nature of those studies. So I can assure you that is a top focus of the team right now.
All righty. Thank you, Chris. Next question. This is a simple, but I think pretty important question. Kristen would like to know, do you need to purchase Benchling compatible scales, et cetera, to utilize this auto data generation?
No. So generally, my short answer to this is that we will work with most modern equipment. And by modern, I mean that if you have a 20-year-old set of balances within the lab, we might run into some difficulties. And I've seen that. I had over 20-year-old skills in my own lab. So it's not like I'm pointing figures there. I've experienced it myself. Any modern devices that have any form of USB connectivity, Bluetooth connectivity, we should be able to find a way to get those to work. Having said that, there are preferred devices that will work out of the box, and we're more than happy to provide a list of what those makes and models are, because we always find that it will make life a little bit easier. For example, we have easily established that we can work out of the box with identification systems such as Rapid Lab or SOMARK, or even other RFID providers. With balances, we typically recommend the O-House devices, but we have users that are using the likes of Mettler Toledo. So we're happy to look at any device and we can sort of advise again on a case-by-case basis.
All right. Next question we have comes from John, who says, we rely on CROs for a subset of our in vivo studies. Do they need a license for the application as well? Just curious what the best practices are here that you've seen.
So I currently, like I mentioned, I work with Cradle. And so they have their own ID with us. And so they, as a company, have one ID. And so all the study techs, I usually work with one, but if somebody's out, their whole team has access using that username. So for example, if I said, Cradle123@cradle.com, this is their email. And so all of their Cradle techs can have the username and password, and they have their own way to go in. So they do not use my username. They don't know my password, any of that. They have their own. And then I specify what capabilities they have. So you can do read and write, just read, just write, that kind of thing. And so I set what they use. So they do have their own. They have their own email that they all use. So I know that Cradle, took those, and it's marked that, the whoever collects that data, it is marked who collects it. So I know that Cradle collected this data. So they do have their own email. It was super easy. I worked with the manager of the vivarium there, and I had it running within a couple hours. So it was super easy. I believe that my team, we're a small team, so we have about 10 usernames that we can use. And so with our team and them, we didn't want to give each member of their team that could possibly be collecting data for us their own username. That would get kind of too convoluted for them. And so just having that overall email for that team worked very well for us. And it's worked great so far. We've been doing a study with them for almost 2 months now, and I've never had an issue, and they're able to get in super easy.
Yeah, just to expand on that, Catherine, I have my own sort of thoughts on the collaborative aspect between CROs and what is essentially their clients, the biotechs and the pharmas that are using their services. I speak to CROs quite a lot, and obviously their challenges are maybe slightly different than might be for a biotech, in that what they spend a lot of their time doing is feeding back to their clients, reporting, and trying to do that in a format that makes sense. And as Catherine has alluded to, what I have always wanted to get to the stage with is the ability for CROs to effectively provide like a live tracking mechanism for their studies. Because I've used CROs in the past, and I knew that I was always waiting for that Friday afternoon email where they sent me the spreadsheet telling me how the study's going. I'm like, paying a lot of money for this study. Like, I should be able to see how it's going at any point in time. So we are having discussions around how that might work, how we might be able to automate collaboration. As I say, I'm deeply interested in this, so I would welcome anyone to contact me directly with thoughts and considerations that you might have there.
Alrighty, thanks for that, guys. Next question we have comes from Kevin, who says, we use Benchling for large molecule only, but studies is good for both large and small molecule work. Is it possible to do cross-database work without dual registration in small molecule source systems as well as Benchling?
Yeah, I can maybe Wendy will have something to add here. I'll sort of talk a little bit around how we see this. So in terms of like being able to access data across databases effectively. So if you had like a Benchling license historically, and then you were to start using studies for your in vivo models, as Wendy showed, we have rolled out some forms of integration there already, largely focused around sample workflows, whereby if you collect samples in the studies platform, that information is instantly accessible within the core Benchling registry. And then you can do things like set up downstream bioanalytical processes. Say you wanted to take those samples and run a PCR. So we do have that connectivity where data is able to be easily sent between the two systems. Obviously, the long-term goal is that we want to provide a full integrated system, and that is something that we have dedicated teams that are working on long-term. And one of the questions that we get asked quite a lot at the minute is pulling data that might be stored within Benchling into the studies platform. So that could be like, say you were storing your cell lines that are going to be used in a tumor study, or just storing your formulations or other things, and being able to access those in a simple drop-down fields within the studies platform, something that we're actively considering right now. I don't know if you have anything to add there, Wendy. I might have covered everything.
Yeah, no, I think you're right. What I'm thinking is that that integration is done. Obviously, the integration between those registry systems can be done directly into Benchling. So it would be kind of one system, and everything will kind of move between Benchling and studies. I mean, being a module from that registry. Mm-hmm.
All right, thanks, guys. Next question we have comes from Danny, who says, do studies have the ability to convert clinical observations data into a SEND format?
Yeah, something that we're actively looking at the minute. So we do have a full clinical observations feature just for transparency. And what that enables you to do is to set up things like custom scoring systems that you're going to be using on an observation by observation basis. So like, say, my lab typically scores barbering on a scale of 1 to 5. We could set that up on a numeric system, or maybe we score like the ulcerated status of the tumor on like a mild, moderate, severe type scoring system. So you can be quite flexible about how you do that, assign the animals in sort of an individual basis. And I think I might have also seen another question which asked about how might veterinarians be able to access this data. So as those health checks and clinical observations are recorded, we do have like a centralized area where they are collectively stored on a study by study basis. So if that could go into a study, say like, show me everything that's been recorded over the last 7 days, and then they could go and physically examine the animals and pass verdict there. In terms of putting it into SEND format to help with reporting, something that we are actively looking at as a team.
All right, thanks for that. This next question, I think, Catherine, you may have touched a little bit on this in your fireside chat, but I think it's an important point too. Shelby would like to know, does studies have an export format option so data can immediately be uploaded into statistical programs such as R or GraphPad, Prism, which I think you may have mentioned you use, SAS, et cetera, something like that?
For me, we mostly use Prism, so I can really only speak to that, but I have had no issues with, downloading the Excel file and importing it into another software and having it read it, as long as you have it in the correct typeface. So, like .exl or that kind of thing, it should read it no problem. Prism it sends directly to that, but I haven't had any issues with downloading the Excel file and then uploading it into another software. Chris might be able to better explain that, but so far I haven't had any issues.
Yeah, we have a number of export formats. The GraphPad Prism one is easily the most popular. And as Catherine says, that will automatically generate a Prism file, so a PZFX file with all of your data tables already formatted. For things like R, we have not built a automated system or export that can get the data directly there. We have other export formats where it could probably be manipulated easily to do that. Also on this topic, one of the benefits of studies is we do have sort of a full end-to-end API. So if there is anyone who is technical, able to write code in any format, or maybe you're lucky enough that within your organization you have your own IT resource, they can get in contact with us. We can talk them through the documentation, how to work with our API, and you could potentially build some of that out to have those automated workflows.
All right, great. I have time for a couple more questions since we have so many from the audience. Bobby has a question. He'd like to know, are multiple scientists able to enter in data for study at the same time?
Yes, one of the benefits of a cloud-based system. So it's accessible by several people at the same time. Do you have any experience with that, Catherine? Would there ever be a situation where you might have yourself and a colleague, or yourself and someone from Cradle who would be working on a study at the same time, or has it typically been one?
So far, it's really only been one, but we do sometimes have that. And the most experience I have with it is, I'm inputting data, and at the same time, my boss is reading. So, that hasn't been an issue at all. He can literally see as I'm doing measurements, and it updates. And same with me looking at Cradle. We haven't had any overlapping inputting studies at the same time. But, they've been doing measurements and I create a new study and there's been no issues with that. So I guess we'll see with, two different studies having measurements put in at the same time. But all the other aspects at the same time have had no issues whatsoever. And multiple people can, be looking at the same study at the same time. Like I said, I usually put read-write restrictions on it. So there's that. But other than that, no issues.
Yeah, and I think you've raised a good point there that would maybe alleviate a fear associated with that, because I certainly know that if I had a study and I was working and all of a sudden data started changing and somebody else had access to it, that would be a bit of a concern. The way that we get around that is we do have study-specific permissions. So in every single study, you can assign the people that you want to have access to that study and who should be able to influence it. So as Catherine's pointed out, you could say that I want to give myself write access, one of my colleagues write access, and maybe, like as you pointed out, like it might be your boss who's sitting in the office and never actually comes into the lab and they just want to glance at the data. Just give them read access. It means that they're never able to change data points anyway.
Yeah, exactly that, Chris. There's only, like I said, I'm the lead for the US. I'm really the only one who should be inputting data on my team, besides the Cradle CRO support. So everyone else has read-only restrictions, just to prevent, any accidental change in data. And I don't know if it's really saved us at all, but with that on, it just gives kind of a peace of mind. So exactly that.
Right. And I think we probably have time for one last question. So Sophia has a question. She says, what are the scheduling capabilities of studies? I'm currently using Outlook to organize the team and send invites with study IDs and animal IDs. Is this something studies could do?
Yes. So there's a couple of aspects to this. So we do have a task management system. Quick overview on how that is structured. So you can set up combinations of different tasks within your study. So for example, really basic example, I could say I want to set up a study. I want to measure the body weights of the animals every 3 days, and I want to measure their tumor size every Monday, Wednesday, and Friday. And maybe I want to scan the animals with an IVIS system on study day 25. So you can combine what I would describe as calendar-based tasks, which happen at regular frequencies. Like think of it as setting up an event within the likes of Outlook or Google Calendar, and combine them with what are known as conditional tasks. And those tasks will only actually trigger when a condition is met. And the example I provided there was like an animal hitting a particular day of the study. The benefit of that is that you could set up that protocol. And if we do have some teams that particularly do a lot of neurology studies, and they'll do rolling recruitment into their study. So just because of that, every animal will effectively be on a different study day on a single calendar day. Like you might have had an animal that's been in the study for 30 days, whereas another animal was only enrolled yesterday and it's on study day 1. And so what we want to be able to do through that task management system is easily identify on any given day which animals have hit that condition. So it's a nice way to do that. You can combine all of those different tasks into what are known as task templates. And those task templates can actually be reused across lots of different studies. So I know that historically, I always measured the body weights, measured the tumor sizes on the same days of the week. I was a creature of habit, and it enables you to easily set that up. We also have a couple of other features that can potentially help with that. we have ways that you can set up, ways that you can sort of connect other systems, send requests, and there are email notifications associated with that. But the task management system is actually one that we're looking at heavily in terms of how that can be integrated into the wider Benchling R&D cloud. So we have some excellent features there, mainly focusing around the workflow feature. And I would like to see how we can tie together a lot of the in vivo study protocol around a sort of workflow that can be set up within the wider Benchling platform.
Yeah, and I will add to that I do use the task management system. If I go in and I have, a number of studies that need to be measured that day, I also do my tumor measurements and body weights the same day, Chris, so you're not alone. But yeah, all those tasks show up on my dashboard. And so I know what I have to do that day, and it just pops up automatically. So at the end of everything, I can go back to that dashboard and make sure that I've, finished everything on my list that day. I do still use Outlook, but it's mostly to block off time in my calendar to make sure I have time to go do those things and allocate that. But the task management system has been great. And I have also allocated, I do certain measurements Monday, Thursday, some Tuesday, Friday, and I've been able to allocate those tasks and create those, like Chris said. And so it's really, very easy once you have that set up. I've used it for multiple different studies, and I just, click on which one I want. And those tasks just show up on my dashboard for that day once I start the study, and it's really easy to use.
Would it help if we provided some form of integration in the future directly to linking up our task system to Outlook?
I'd say it wouldn't hurt. And it's worth a shot. I use Outlook, especially because I also want my boss to be able to know when I'm doing certain measurements if we're on a time course study. And so I can invite him to that so he can look at a certain time. And so if we're able to integrate it, I think that would be fantastic. And it can kind of, instead of two platforms, just use one. But it's also my own personal preference, as I've been doing Outlook for years, using that. And so I think going into studies and being able to see my tasks for the day is enough for me for now. But yeah.
All right, guys. Well, thanks so much again. This has been a great webinar. Chris, Wendy, Catherine, thanks so much for your great presentations. So I'd like to remind everyone that the webinar will be archived on the Gen website at genengnews.com for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward the link to any of your friends and colleagues, which we always recommend. Again, I'd like to thank Wendy, Chris, and Catherine for great presentations. And I'd like to thank you, the audience, for your attention and very thoughtful questions. And a very special thanks to Benchling for sponsoring this webinar. Hopefully, we'll see you again at another Gen webinar in the near future. Goodbye for now. Everyone stay safe and healthy.